Just the ordinary surrounding world. Somewhere it's calm and peaceful, and somewhere else busy and bursting with surprises.
There are around 1 million of bacteria in just this little amount of ground, but there are also some viruses. Same as humans, bacteria have an immune system protecting them from infections. And in the next 3 minutes I will talk about how this system is being used for curing people's diseases. So my topic is CRISPR-Cas9 systems. You know, the structure of organisms is being coded by genes, and those are constructed from amino acids that are made of nucleotides. Some sequences of genetic code are complementary to each other, and this is the key to the work of bacteria's immune system. Not long ago, a strange part of genetic code of bacteria was found. At first glance, it was useless and was not coding anything. However, this first impression was wrong. After some investigation, CRISPR clustered regularly interspaced short palindromic repeats were found. Those are completely identical sequences of nucleotides placed in the mysterious useless part of DNA. The parts of DNA between CRISPR are not identical. They are called spacers. A lot of the spacers, as was soon uncovered, are similar to the parts of genome of different viruses. Those parts of DNA are being used to produce CRISPR RNA. Near the CRISPR part of DNA there is a Cas, CRISPR-associated sequence, locus. This is a matrix for the synthesis of Cas proteins.
So what do we have now? CRISPR RNA, which contains sequences complementary to the parts of virus genome, is being produced using the CRISPR part of DNA. And Cas proteins are being produced using Cas locus. Produced RNA and proteins are being used for creating complexes. If a virus somehow gets inside of the organism, it's found by those complexes using the complementary parts of the code. After that, the complex attracts other Cas proteins which take the leading role, kind of like genetic scissors. They ruin the recognized virus DNA.
Some bacteria use only one but huge Cas protein, Cas9, which can do the functions of all other Cas proteins in a time. Later it was proved that CRISPR-Cas9 can work not only in bacteria cells, but in cells of mammals too. And this is the point where scientists thought like, hey, they ruin all DNA that they recognize, so what if we can make them recognize the damaged part of human genes instead of virus DNA? So scientists modify the bacteria's DNA. Instead of parts identical to those of virus, they place there the lab-produced sequences, which are similar to the damaged human gene. After that, upgraded DNA is placed into an ill organism. Recognizing the damaged gene as a virus, Cas9 protein ruins it. That's how the damaged part of genes can be removed. This means that soon we will be able to cure people who were born blind, deaf, having genetic illnesses like hemophilia and lots more. By the way, some blind mice were already cured using this method.
Imagine how fantastic would it be to enable a blind man to see. I'm Sasha and thank you for your attention.